introduction and for the opportunity to be here today and talk to you about some of the work we've been doing around rare diseases in the Middle East and mainly in Dubai. So I'm sure many of you are aware, who are especially working on the diagnosis for rare diseases. Currently, the diagnostic yield, which is the proportion of patients who receive a positive report in rare diseases using the current state of testing, is around 40%, which means that around 60% of the patients yet do not get an answer. You could argue that some of those patients might not have a genetic or a monogenic cause, but we know there are knowledge, technological and methodological gaps that need to be filled to close down this diagnostic gap. And of course, you know where I'm headed with this. Of course, one of the first things that comes to mind is about sequencing technologies. Short-read sequencing has been great in identifying SNVs and indels, which has been driving the diagnostic yield that is now maxed out at around 40%. But we know that long-read sequencing, such as Oxford Nanopore, adds another rich layer of genomic variations, including repeat expansions, copy number variants, structural variants, as well as epigenetic DNA methylation signatures that could enhance the diagnostic yield. And the question is how much of this actually contributes to the diagnosis of patients with rare disorders, and if that is possible, could long-read sequencing be the only clinical genetic test in diagnostic labs? So we set out to answer this question, and our study design included 53 patients, 14 of whom had positive pathogenic variants, including SNVs, indels, copy number variants, chromosomal aneuploidies, methylation defects. Of course, these were tested by different technologies in a clinical lab, as is currently the state of testing. We used this cohort to establish and optimize a protocol and pipelines for calling all these variant types. And then taking this protocol and pipelines, we applied it into another cohort who were negative by short-read sequencing and asked how many of those patients would now become positive had they been tested by long-read sequencing. And of course, we started with a protocol, wet bench protocol optimization to get enough good quality data for, of course, variant calling, and for SNVs, copy numbers or structural variants. We use published as well as in-house scripts for variant calling, annotation, and filtration. Keep in mind, this is not a discovery cohort. We're talking about a clinical yield, so we're focusing on things that would have been missed in disease genes. So we have to come up with certain strategies when we're doing this on a clinical basis to trim down thousands of variants to a minimum number that we can actually curate manually and report. We've also extracted the episignature profiles for the known 42 diseases that's currently being offered on EpiSign or the Illumina arrays platform. We call this epimarker. And we have identified what we think is a novel SMN1 specific methylation that we added to this epimarker, so now it includes 43 diseases. This is just showing you our funnel down or filtration strategy for copy number and structural variants. And like I said, this is not a discovery cohort, so we're focused on things that affect the gene and mainly disease genes. And we're also looking at events that are unique to each one of those patients. So it's not the most sensitive, and we're likely missing things, but we're focusing on the lowest hanging fruit. And it is specific or highly sensitive, at least for the positive cohort that we use, because we've identified all of them using this filtration strategy. Here's an example of a deletion, inverted tandem deletion duplication shown in green by Oxford Nanopore, which was originally identified by chromosomal microarrays. And of course, we ran positive samples for the epimarker. And an example here is an Angelman syndrome. And as you know, individuals usually have biparental inheritance at 15q11.2, as shown in the top IGV, with blue and red methylation marks. And the middle lane has a patient with Angelman syndrome, which the epimarker can identified using this heat map. You're looking at the first patient that doesn't have methylation at this position compared to others, and as quantified in the bar graph here. However, when you have an Angelman disease, the mechanism could vary, and it could be due to a deletion or epimutation, or could be due to UPD. So we could extract this information from long-read sequencing, and as you can see, this patient has loss of heterozygosity on chromosome 15 compared to other chromosomes, suggesting that this is a paternal uniparental disomy. It's a male, so the X chromosome serves as an internal control, as we can only see homozygous variants in this chromosome. And this is the SMA profile that I mentioned to you earlier. I'm not sure you can see this. The top two IGV viewers is looking at the lack of methylation from exon 7 all the way to the end of the gene. The second two IGV views are from carriers who have 50% of the methylation at this region compared to the last one, which has the full methylation profile. This is also seen on the heat map from epimarker. So you're seeing no methylation in two positive controls, almost 50% of the methylation in carriers who have one copy of SMN1 compared to the full methylation in other control samples, and this is quantified here. So now we have all this protocol and package. We wanted to apply this to a negative cohort of patients, which in our setting are mostly Arabs and from the Middle East. The main presentation is neurology or neurodevelopment disorders, and we were targeting an average coverage of 30x and an N50 of 12 kb. Here's an example again of the funnel down approach going from thousands of SNVs and CNVs and SVs to a handful. This is also shown here on the right. You're seeing the number of variants that are being called by different variant callers. But then on the left, using the filtration strategy, it kind of trims down the number of variants that we look at per patient. And the things in orange are what we think are pathogenic variants that now turn those cases into diagnoses. In addition, we identified two methylation defects that are also likely to explain the diagnosis for this negative cohort. Let me give you a few examples. The first patient who presented basically with anterior segment dysgenesis had exome sequencing that was inconclusive because they had a single heterozygous frame shift variant in SLC38A8. This gene causes autosomal recessive anterior segment dysgenesis, so matches the patient phenotype, but of course the report is inconclusive because it's heterozygous and the disease is recessive. With long-read sequencing, we identified a deletion shown under the red bar over there. You can appreciate the decrease in coverage, the break point, and the loss of heterozygosity. And of course, using the long read, we could phase this. So you could see that on haplotype 2, there are no reads around this region, which suggests that the deletion actually is in haplotype 2, while the frame shift on the other side actually is in haplotype 1. So this confirms the compound heterozygosity and a diagnosis for this patient, which was confirmed by chromosomal microarrays and PCR-based fragment analysis. In the second case, we identified a homozygous partial deletion of the 3' UTR of the gene MPLKIP, which causes learning disability and distinctive brittle hair, which was actually very unique for our patient, suggesting it's likely to explain this patient's phenotype. Of course, deletion of 3' UTR are hard to interpret, but if you remember, 3' UTRs could be regulated by microRNAs, could be downregulated transcriptionally or translationally. So we did transcriptomic sequencing and actually found that this gene was one of the outliers and is highly overexpressed compared to other controls. And this was actually done independently. So when we set out to do this project, we also wanted to see how much RNA sequencing contributes to the diagnostic yield. And this gene was one of the main outliers in this patient. So it's nice to kind of see the correlation between the homozygous deletion as well as the transcriptomic data. The third patient has a methylation profile that was consistent with this disorder, Hunter-McAlpine syndrome, which explains the patient's phenotype. And independently, we also found a duplication of the NSD1 gene. So this disease has been characterized by this methylation profile or a duplication of the NSD1 gene. And in our case, we're identifying both of them, and we think the duplication is actually what's driving the methylation profile. If you're familiar with this, NSD1 deletion actually causes Sotos syndrome. So the opposite of this disorder would be a deletion of NSD1 as well as hypomethylation. In our case, we're seeing hypermethylation and duplication of the gene. Of course, this was confirmed by chromosomal microarrays. And the last case I want to show you here is a patient who's 14 years old, undiagnosed, and we identified this lack of methylation in this SMN1 region. As you can see in the second IGV viewer, this is actually a patient or an individual with 3 SMN1 copies. And I hope you can appreciate that the methylation is more pronounced compared to the lowest 2-copy number SMN1. So not only that this methylation is specific to SMN1, but seems to be also dose-dependent, which helps in kind of quantifying the copy numbers in patients with SMA. Of course, the heat map is shown here as well for the patient and a positive control as well as 2 carriers and negatives. And we confirmed this by droplet digital PCR. So overall, out of the 39 negative patients, we identified 5 that were positive, because one of the patients had a CNV as well as a methylation defect. for an overall diagnostic yield or increment of 13%. If you're interested to know more about our study, it's now in a preprint. I know we have limitations, including the smaller size of the cohort, but recently, I think a week ago, this larger study was actually available in preprint using hi-fi long-read sequencing on a larger cohort, and the magic number is 13% diagnostic yield. So I think we're hitting around the same diagnostic increment, at least using current bioinformatic tools. In addition to increasing the yield, of course, it's really interesting. We set ourselves not to do any discovery, but it's really unavoidable. So we identified this SMN specific methylation, which has both diagnostic and therapeutic implications. As you know, for those who work on SMA, it's really hard to distinguish SMN1 from SMN2, but finding a methylation mark specific to SMN1 can be used as a diagnostic marker. But also this region that's methylated is actually what distinguishes SMN1 from SMN2. And it's exon 7 that's expressed in SMN1 and not SMN2. So one could contemplate that if you introduce this methylation SMN2, you could drive exon 7 expression and rescue patients with the disease. And that's really interesting if you think about it, because SMN1 is actually an RNA binding protein and has a lot of functions in RNA biogenesis. So I hope I presented data to convince you that long-read sequencing not only enhances the diagnosis of patients with rare diseases, but also leads to novel variation discovery, which has implications for diagnostics as well as potential therapeutics, and also helps us kind of gain more insights on the biology and disease mechanisms for some of those rare diseases, including a classic disorder like SMA. I would like to finish by thanking the team that's behind all this work, and especially Shruti, who's the bioinformatics scientist who's built all the pipelines. Of course, Nanopore for their continuous support, and all the patients who consented for the project. Thank you so much.